I want to thank the organizers, thank all of you for being here late this afternoon, where I have an opportunity to provide an update on and overview of WindMIL Therapeutics, a company that is built around a straightforward but we believe fundamentally important concept, and that is that in cell therapy, cell source matters. If I just give you a quick overview of the history of WindMIL Therapeutics, the mission of the company is to translate novel insights in bone marrow immunology into life-saving therapies for patients with cancer. The company was founded in early 2016 based on technology that was taken out of Johns Hopkins University. The technology is based on bone marrow-derived T cells, which are naturally antigen-specific and contain a very high percentage of memory T cells. Those cells which we produce as marrow-infiltrating lymphocytes, or MILs, are produced through a proprietary process that activates and expands these bone marrow-derived cells. We have a very strong intellectual property position, trade secret, and know-how which surrounds our MILs technology and provides an exclusive position in terms of manufacturing and development. We have, as you'll see in a moment, an accelerated clinical development plan that we think drives rapid value creation and a highly experienced management team. To come back to the idea that in cell therapy, cell source matters, this graphic contrasts the characteristics of MILs, our product, with T cells that are derived from other sources in the body. T cells derived from the tumor site typically are of an exhausted cell type. Those that are derived from peripheral blood, which are really the majority of T cells used in adoptive T cell therapy today, are not broadly antigen specific. In contrast, our cells, those that are derived from the bone marrow, are patient specific, they're broadly antigen specific, they're highly cytotoxic, and they're highly persistent. And WindMIL is the only company that can activate and expand this source of tumor-specific memory T cells that are found in the bone marrow. Just to give you a quick overview of our manufacturing process, it's actually, again, very simple and straightforward. Bone marrow aspirate is harvested from the cancer patient. It is transported to our manufacturing partner, Cognate Bioservices, where the aspirate undergoes a 7 to 10 day proprietary process through which MILs are activated, expanded, release tested, cryopreserved, and then sent back to the treatment site where this cellular product that is enriched for memory T cells with tumor antigen specificity is infused back into the patient. Our strategy in one slide, we are a company that is deep into clinical development at this point. We're using that platform of MILs technology to develop opportunities in 3 clinical spaces. On the left, hematologic malignancies, where we've completed, through our partner Johns Hopkins, 2 phase 1 studies and 1 phase 2 trial. That has involved MILs being administered already in the clinic to more than 100 patients. We've seen encouraging efficacy in phase 1. We've seen a very favorable safety profile, and we have phase 2 proof of concept data in high-risk multiple myeloma that will be available late this year or early next year. The second area of opportunity we're pursuing is in solid tumors. There, our initial target is the second-line treatment of non-small cell lung cancer, an area of very high unmet medical need, and we think an accelerated development opportunity. We have a phase 2 trial that is underway. We expect first patient, first dose in the 4th quarter of this year, and we're currently planning studies in other solid tumors for late 2020 or early 2021. And the third area that we're pursuing clinically is gene-modified MILs, what we refer to as CAR MILs. We believe CAR MILs represent a potential step change in gene-modified T cell therapy, and perhaps are uniquely positioned to address some of the limitations of current CAR T cell therapy. There, we're pursuing a partnering strategy, looking for that partner to provide CAR technology and experience, access to unique CAR constructs, and an opportunity to get into the clinic quickly and produce clinical data with our CAR mills. And we're hopeful that in the late fourth quarter of this year, early first quarter of next, we'll be able to announce a partnership in that regard. We're deep into discussions in that partnership. Just a little detail around each of the areas. First, hematologic malignancies. As I said, there have been 2 phase 1 trials completed. A phase 2 trial is actually complete, and the efficacy data will be reported out late this year, early next. A phase 1 trial in high-risk multiple myeloma patients, 22 patients, difficult to treat. What we saw was a 59% response rate, 86% of patients with stable disease. Very impressive in that patient population. The success in that trial, again at Hopkins, led to a phase 2 trial. 90 patients post-transplant were randomized in a 2-to-1 randomization to either Mills plus Revlimid or Revlimid only. The primary endpoint of that trial is progression-free survival. We know the safety profile is good. Again, we are looking forward to seeing the efficacy data from that trial late this year or early next. And the final study on this slide is actually what might be described as an exploratory trial, but it's quite interesting. These are patients that were allotransplant patients. Mills derived from the bone marrow of these patients post-allotransplant. And the data, again, very favorable safety profile, no GVHD, good responses, 14 patients, it's a limited population. But we're looking at this as a possible alternative to donor lymphocyte infusion in the post-relapse treatment of these patients. If I turn to solid tumors, preclinically there is good reason to believe that we'll see Mills efficacy in solid tumors. We know that immune models indicate that a single antigen-specific encounter generates a population of memory T cells. And these memory T cells home to the bone marrow and they actually persist there for extended periods of time. And our research and the research of others has shown that these tumor-specific T cells exist in such solid tumors as non-small cell lung cancer, prostate cancer, melanoma, breast cancer, and other tumor types. And building on this preclinical experience, we had filed an IND with the FDA in May of this year. That was accepted by the agency in June to begin a phase 2A trial in second-line non-small cell lung cancer. Part of the data that supported that was this preclinical work demonstrating the existence of tumor-specific T cells, MILs, in lung cancer patients. In this preclinical work, bone marrow was obtained from patients with lung cancer, 4 patients shown on this slide. It was activated and expanded per our proprietary process, and then presented to essentially media, myeloma lysate, and lung cancer lysate. The myeloma lysate was a negative control, and the data demonstrate the clear ability of Mills to specifically recognize antigens in that lung cancer lysate. The trial that we're going to begin, and as I said, first patient, first dose will be in the fourth quarter of this year, is a trial where patients who are relapsed or refractory to PD-1 inhibitors will the first 3 to 6 patients will be Mills only, intended to clearly demonstrate safety, after which we'll move into a combination regimen of Mills plus PD-1 inhibitors in 20 patients. It's an adaptive trial design. The primary endpoint is response rate. And with that adaptive design, we have the ability to expand this trial quickly and easily. And we think provide the basis for a very rapid development timeline in non-small cell lung cancer. So we'll see the data from the 3 Mills only patients in the first half of next year, and begin to see data from the Mills plus PD-1 inhibitor patients in the early part of 2021. To turn to that third vertical, what we call CAR mills. CAR T therapy has been a very promising advance, but clearly limitations exist. Antigen escape variants, suboptimal trafficking of the CAR T therapy in the tumor microenvironment, and a lack of persistence. There are a variety of approaches people are taking to try and address those limitations. Engineering more CARs into the cells, genetically modifying to drive them to the tumor, and other approaches. We actually believe the solution is in the nature of the T cell itself. CAR-MILs, we think, represent a step forward and perhaps a step change in genetically modified T cell therapy. And that's for 4 reasons. If you look at the MILs themselves, our work preclinically has demonstrated that they retain their non-CAR-based tumor specificity through their native TCR. And we see that both after transduction and after CAR engagement. And they can recognize tumor even after the CAR's cognate antigen is lost. They're still there to kill tumor. Secondly, they home to the tumor site via increased CXCR4 expression. We know that the stroma of solid tumors expresses SDF1, and that acts like a beacon for MILs, attracting them to the solid tumor site. Third, we've shown preclinically that at much lower effector to target ratios, CAR-MILs are more effective than CAR-PBL essentially in killing. They're more active. And that's particularly true with repeated challenge. You see the gap between the 2 types of cells widen as you repeat the challenge. And finally, we know that MILs persist over the long term. They're detectable in those early phase 1 patients in high-risk multiple myeloma. We've seen MILs out to 8 years, and it's not at that point because they cease to exist, we just stopped looking. But we know they persist for an extended period of time. We believe also that CAR-MILs offer us a path for accelerated development. As I said, this is an opportunity we intend to pursue through partnership. Our initial target will be hematologic malignancies, and we think there's the potential, after some initial preclinical work, to move quickly into the clinic and begin to show comparative efficacy data versus established CAR T therapy by the early part of 2022. We have a strong, experienced management team pursuing these opportunities. We're a company of only 20 employees, but this management team is deeply experienced and highly capable in the areas that matter to advance the opportunities that we see in front of us at WindMIL. And we're also provided insight by a distinguished scientific advisory board. If you look down and across this roster, we have experts in hematologic malignancies, in solid tumors, and in genetically modified T-cell therapy. They're very active, they're very engaged, and they're continuously providing insight into the path that we follow at WindMIL. So in summary, WindMIL is the only company that can activate and expand a natural source of tumor-specific memory T cells that are found in the bone marrow. Very deep know-how and a strong intellectual property position provides us with an exclusive position to drive MILs development and commercialization. To underscore that point, if you think about the tens, if not more than 100 companies that are involved in cell therapy today, WindMIL is the only company doing work with MILs. Extensive preclinical research confirms the unique profile of MILs, and clinical study data across those 3 studies that I mentioned earlier in more than 100 patients have shown very favorable safety and, we believe, promising efficacy in hematologic malignancies. We expect the phase 2 data from that 90-patient trial in high-risk multiple myeloma to be available late this quarter or early next. We are cleared to start a phase 2 trial in second-line non-small cell lung cancer. First patient, first dose in that study will be in the 4th quarter of this year. And finally, we're in active discussions with partners to develop clinical proof of concept for what we call CAR-MILs in a comparative clinical trial with existing CAR T therapy. And we would hope to have data from that study beginning in the early part of 2022. And finally, the profile of the company and the rate at which it's accelerating is illustrated on this slide. 3 very important milestones in the 4th quarter of this year, as I just mentioned. Mono MILs therapy data in non-small cell lung cancer beginning the middle of next year. The combination data, the first 10 patients response rate available in the early part of 2021, and then, as I mentioned, in early 2022, the CAR-MIL data. So I thank you for your time and attention, and I'm available for questions after the presentation. Thank you.